So in the early 1930s, there was a scientific desire to visualize the fine details of the interior structure of organic cells like the nucleus and mitochondria. So before that, the the microscope was become the light microscope was there, but there was need to have more magnification. Uh, for that, electron microscopy was okay. And so electron microscopes are scientific instruments that use a beam of electrons instead of light, so to examine objects on very fine scale. And after that, the image produced by these electron microscopes is perceived by CRT, that is cathode ray tube, that contains one or more electron guns. And the CRT, and that is more famous for like the people who have the old television. They have the at the back of that screen there is an extension, and that extension is actually cathode ray tube. We will be talking about this in detail in the coming slide. And a magnification of 10,000x plus was achieved, which was not possible using optical microscopes. And this is actually the cathode ray tube using electromagnetic focus and deflection. If you can see, this is the heater, and this is the control grid that controls the heat, and the cathode is there, and emission of electrons, and this is the anodes, and these are electron beam, and these are the focusing coils, and these are deflecting coils, and the fluorescent screen. And they are perceived here on the screen. The beam is perceived here on the screen. So if you can see some another like perspective of the same thing, like CRT, this is deflection coils. The one is here, the deflection coil. These are the deflection coils, like shown here. This is another representation of the same. And the electron beam, here the electron beam is shown. Like here, this is the source of that, and this is the electron beam, and same is shown here. And after that, that focusing, this is the focusing coil here, focusing coil. And here you can see the focusing coil is represented here. And same is phosphor layer on the inner side of the screen. And is this one. So that is phosphor layer on the inner side of the screen, and it's a substance that exhibits phenomenon of luminance. It emits light when exposed to some type of radiant energy. So this will emit light when exposed to some type of energy. So this is phosphor layer on the inner side of the screen, and it will emit light when struck by the electron beam. So it will emit light. The screen will emit light. And after that, the filament for heating the cathode, and that is this one, this thing. And here is shown here in and here. So and is this number 6 one is the graphite layer on the inner side of the tube. Here, the graphite layer on the inner side of the tube. So here it's is given here. So this is the graphite layer on the inner side of the tube, and after that, rubber or silicon gasket, where anode voltage wires enter the tube, anode cup. So here is the 7 one, here is 7 here. So the anode cup, and a cathode is here. Cathode is here, and this is the cathode to emit the electrons, and airtight glass body of the tube. Then the screen is there, coils in the yoke, and control electrode regulating the intensity of the electron beam, and thereby the light emitted from the phosphor. And that is number 12 here. So that control the electrode, and that's regulate the intensity of the electron beam. So this is important. This control the intensity of this electron beam. And after that, contact pins for electrode. So contact pins for, these are the contact pins for the electrode. And we will be talking about all this stuff in detail, but this is just the intro of all these. So these are the contact pins for the electrode, filaments, and control electrode. So and after that, wires, a wire for anode high voltage. So this is the wire for anode high voltage, and the only visible difference are the single electron gun, the uniform white phosphor coating, which is phosphor coating, and the lack of shadow mask. We will be talking about this in detail, don't you worry for that. And this is actually the practical example what do we have previously at our homes. And that is this is the rear of 14-inch color cathode ray tube. This the same cathode ray tube. This is like this one, the same. So this is showing its deflection coils and electron gun. So this was actually, we have used these things at our homes, um, in our TVs now, that these things are replaced with LEDs and LCDs, etc. We have a video for the intro of electron microscopy. Let's check out that, and we will be going in detail in all this stuff. So, okay.
Light and electron microscopes. The beauty of biology and all that we know today is due to the invention of microscopes. Microscopes had been available since the beginning of the 17th century, but a lot of improvements were made in the quality of glass lenses in the early 19th century. The branch of biology dealing with
आपको आवाज आ रही है?
Yes, sir.
Yes, sir.
आ रही है.
design, slide preparation and examination is called cytology. Light microscopy. The light microscope uses light as a source of radiation and has shown us inside cells. It shows us simple structures like the nucleus, the cell wall, and the cell membrane. A light microscope is still used in basic studies in schools today. Electron microscopy. Then came the electron microscopes, which made our microscopic world more detailed. Due to the electron microscopes, we not only saw the cell wall, the nucleus, and the cytoplasm, but we also saw the more minute organelles, like the mitochondria, endoplasmic reticulum, and the Golgi apparatus. The light microscopes were doing great, but no matter how much their designs improved, there was an extent to which the light microscopes could not show. Anything smaller than 200 nm could not be seen. Electrons are subatomic particles that have a negative electric charge and are found surrounding the nucleus of an atom, called neutron. When a metal becomes very hot, some of its electrons gain so much energy that they escape from their orbits, like shooting stars. The energy in free electrons is associated with the proportion of waves. The higher the energy, the shorter the wavelength. Electrons are a very suitable form of radiation for a microscope for two major reasons. Firstly, the wavelength is extremely short, and secondly, because they are negatively charged, they can be focused easily using electromagnets. The magnet can be made to alter the path of a beam, similar to how a glass lens made to bend the light. There are two types of electron microscopes in use today: the transmission electron microscope, TEM, and the scanning electron microscope, SEM. In the transmission electron microscope, the beam of electrons is passed through the specimen before being viewed. Only those electrons that are transmitted are seen. This enables us to see different components inside the cells. In a scanning electron microscope, the electron beam is used to scan the surfaces of structures, and only the reflected beam is observed. The advantage of this is that surface structure, for example, like the chitinous outer body of insects, can be seen in great detail before being viewed. In the transmission electron microscope, the beam of electrons is passed through the specimen before being viewed. Only those electrons that are transmitted are seen. This enables us to see different components inside the cells. In a scanning electron microscope, the electron beam is used to scan the surfaces of structures, and only the reflected beam is observed.
डिफरेंस का पता चल गया सेम और टेम में?
यस सर।
Yes, sir.
सेम में क्या था? टेम में, सॉरी, टेम में
सर टैब में वो ऑर्गेनिज्म के उसमें सर विजिबल होने से पहले पास करती है, उसके बाद हमारे पास जो लाइट सोर्स पहुंचता है तो हम पूरे सेल के डिफरेंट पार्ट्स को इजीली जो है देख सकते हैं। सैम में सर वो सिर्फ सरफेस को फोकस कर रही होती है।
ठीक है, एग्जैक्टली इसी तरह है, बिल्कुल। एक दफ़ा जो कवर नहीं किया हुआ बस किसी ने मिस कर दिया तो एक दफ़ा देख लें।
TEM and the scanning electron microscope, SEM. In the transmission electron microscope, the beam of electrons is passed through the specimen before being viewed. Only those electrons that are transmitted are seen. This enables us to see different components inside the cells. In a scanning electron microscope, the electron beam is used to scan the surfaces of structures, and only the reflected beam is observed. The advantage of this is that surface structure, for example, like the chitinous outer body of insects, can be seen in great detail.
ठीक हो गया
सर। सर जो स्कैनिंग इलेक्ट्रॉन माइक्रोस्कोप है उसके अंदर जो इलेक्ट्रॉन की बीम की जो पावर होती है उसको स्लो रखते हैं ताकि वो सरफेस के ऊपर ही रिफ्लेक्ट बैक हो जाए। ये सर दोनों के ऊपर हम स्पेसिमेन के ऊपर ही अपनी इलेक्ट्रॉन बर्बाद करें, लेकिन एक के अंदर पास थ्रू हो रही है, एक के अंदर नहीं हो रही।
हाँ, वो असल में डिपेंड कर रहा है कि हम उसको ले कहाँ रहे हैं। अगर इसी वीडियो का अगर देख लें तो स्पेसिमेन को बिल्कुल लास्ट में रखते हैं और स्पेसिमेन के उसके बाद लाइट आगे पास नहीं हो रही, ठीक है। और फर्स्ट वाले केस में क्या था कि वो उससे पास हो रही और वो जो डिटेक्टर है वो उसके नीचे लगा हुआ है, ठीक है। तो
ओके
एक में
सर।
रिफ्लेक्शन है और एक में ट्रांसमिशन है, मींस के
सर
उससे
एक और भी है सर।
जी 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 जी
सर ट्रांसमिशन में हम जो लाइट है उसको फोकस करके एक जगह से पास कर रहे होते हैं, लेकिन उसमें स्कैनिंग में हम स्प्रेड आउट लाइट
स्कैटर्ड
यानी कि
करते
हम
होते हैं,
फोकस नहीं कर रहे होते,
स्कैटर्ड कर रहे
स्कैटर
होते हैं।
कर
हाँ,
रहे होते
वो देखते हैं, वो उसके बाद सारी डिटेल्स के हैं, लेकिन फिलहाल अभी ये उतना समझने के लिए काफ़ी है कि लाइक एक से हम ट्रांसमिट कर रहे होते हैं और दूसरे में हम उससे रिफ्लेक्ट करवा रहे होते हैं। अजीम यू आर राइट बाय द वे, ठीक है। Thanks Shafiq for pointing it out here. And next is okay. So electron microscopy components क्या होंगे? Like electron optical column होगा उसमें that consists of electron source to produce electrons. Magnetic lenses will be here. Magnetic coils to control and modify the beam. And after that, aperture to define the beam, prevent electron spray. Like it will prevent the spray of electron, like it will not scatter electron at the start of the emission. So this is how the electron optical column consists of and what it actually constitutes of. And there is another component, and that component is vacuum system. So the first of the component is electron optical column. The second one is the vacuum system. This is common for all for both types of the um, like the electron microscopy. And in the vacuum system, that consists of chamber that holds vacuum. So the first of its function when holding the vacuum, and the second is to pump produce vacuum. So it will produce vacuum, also it will hold the vacuum. And there will be valves in the vacuum to control vacuum and gauges to monitor vacuum. So it's well controlled as well as monitor the vacuum. And the signal detection and display, and that will be consist of detectors which collect the signals and the system to produce an image from the signal. And basically, all these three types of the components, they are the part of either the scanning or the transmission electron microscopy. And these are actually the two basic types of the transmission, the electron microscopy, the scanning and the transmission. So SAM and TEM. We have one another video, a bit more detail, and we will be talking about this. Okay, let's check it out.
He knows a lot about the science stuff, Professor Dave explains.
We just learned all about light microscopy, which was the standard until the middle of the 20th century and remains tremendously useful today. But as we began to learn more about the microscopic world, using light microscopes that could visualize individual cells, we started to become curious about structures that are smaller than a cell, some details of which were difficult to visualize and study under standard light microscopes. Fortunately, electron microscopy, developed in the 1930s and 40s, proved to be much more powerful and allowed scientists to view these smaller structures. Some electron microscopy techniques employ a resolution of 0.2 nanometers, which is more than 1,000 times better than light microscopy. With this higher resolution, electron microscopes are able to magnify their specimen up to 1 million times their original size, allowing researchers to study proteins inside of cells and the molecular structure of viruses, all of which led to a better understanding of the microscopic world. Of course, we have to ask, how do they do this? Well, the answer is in the name. They use beams of electrons, which interact with the sample as the beam is transmitted through the specimen. As we recall from our study of chemistry, electrons are stable subatomic particles that carry a negative charge. Electron beams have a much shorter wavelength than the visible light that light microscopes use, which we refer to as the de Broglie wavelength, as we learned in the Modern Physics series. This shorter wavelength allows for a higher level of detail and resolution in imaging. One type of electron microscopy is called transmission electron microscopy, or TEM. For TEM, specimens are fixed on a support grid and either embedded in resin or stained with a material like uranyl acetate or compounds of heavy metals like osmium tetroxide. Certain structures within the specimen will take up more of the stain than other parts of the specimen, and those sections are called electron-dense regions. When electrons pass through electron-dense regions, they lose more energy compared to when they pass through sparser regions. This phenomenon is used by TEM to visualize the specimen at high levels of resolution and magnification. For the stained specimen to be viewed by TEM, it must be dehydrated, embedded in plastic resin, and cut into ultra-thin slices with a glass or diamond knife. The sections must be ultra-thin so that the electrons can pass through them. When the specimen is secured in the specimen holder, an electron beam is generated and shot out of the electron gun. The electron beam is accelerated down the column of the microscope after passing through the anode. The electron beam then passes through the specimen, once again losing energy as electrons pass through electron-dense regions. The electrons then pass through the objective lens, magnifying the image, before the electrons pass through the projector, or ocular lens, which magnifies the image further and projects it onto the fluorescent screen. This is where we start translating the electrons into an image. The fluorescent screen is coated with a chemical which appears as bright spots when electrons come into contact with it. The intensity of the bright spot depends on how much energy the electrons have when they hit the fluorescent screen. The camera under the fluorescent screen then captures the image made from the bright spots, which is displayed on a computer screen. Transmission electron microscopes are used to visualize structures that are too small to study with light microscopes, such as the cell organelles, proteins, cytoskeletal filaments, and the components of cell membranes. But even with the unmatched resolution and magnification abilities of TEM, it still does have its limitations. Some of these include the fact that you cannot study living specimens or image 3D structures, because the specimens must be dehydrated and cut into ultra-thin slices before imaging. The second type of electron microscopy is scanning electron microscopy, or SEM. With SEM, specimens are coated with vaporized gold or palladium ions, because when electrons come into contact with this coating, they cause atoms on the surface of the specimen to emit electrons. These emitted electrons are called secondary electrons, and these are what SEMs use to visualize their specimen. After the coated specimen is secured on the stage of the SEM, the process starts out similarly to TEM in that an electron beam is shot out and accelerated towards the specimen. In the SEM, the electron beam then sweeps or scans the surface of the specimen, which causes it to discharge secondary electrons. There are two types of detectors in the sample chamber: the secondary electron detector and the backscatter electron detector. The backscatter electron detector picks up backscatter electrons that come from deeper regions of the sample that are a result of elastic interactions between the beam and the sample. The results from this detector are primarily used to look at crystallography and the magnetic field of the sample, but for the purposes of microbiology, researchers focus on the detection of secondary electrons, which allow them to visualize the topography of the sample, or the distribution of parts and features on the surface of the sample. The detectors convert the electron signal into a light signal that is amplified by passing through a photomultiplier before the final image is projected on a computer screen. SEM imaging is primarily used to study the topography of cells and other similar structures. With this type of microscopy, researchers are able to investigate the depth of structures, which isn't possible with TEM, as SEM produces a 3D image opposed to the flat image from TEM. Yet, just like TEM, SEM cannot visualize live specimens, and SEM has worse resolution than TEM. Both TEM and SEM are expensive and require a lot of training, so they are not likely to be used apart from those pursuing a microbiology career. We covered quite a lot in this tutorial, so let's briefly summarize. Electron microscopy has around 1,000 times better resolution than light microscopy, because electron microscopy uses electron beams instead of light, which have a shorter wavelength. This allows some electron microscopes to visualize specimens at 1 million times their original size. Transmission electron microscopy, or TEM, records the differing energies that electrons possess after they pass through a specimen stained with heavy metals to visualize small, detailed structures, like proteins inside of cells, and molecules associated with viral particles. Scanning electron microscopy, or SEM, captures the secondary electrons that are emitted from the surface of samples coated in vaporized gold or palladium ions to visualize the topography of the specimen. And the main drawback, as we mentioned, is that both SEM and TEM cannot be used to visualize living specimens, and they are also quite expensive and complicated techniques to use. And with that, we know a bit about electron microscopy. Thanks for watching, guys. Subscribe to my channel for more tutorials. Support me on Patreon so I can keep making content. And as always, feel free to email me, ProfessorDaveExplains@gmail.com.
Who will explain? Like अभी तक जो पढ़ा है बस अपने words में कोई explain कर दें. जी दुआ प्लीज.
सर हमने इस लेक्चर में देखा कि फर्स्ट ऑफ ऑल तो इलेक्ट्रॉन माइक्रोस्कोप लाइट माइक्रोस्कोप से बेटर किस तरीके से होती है, क्योंकि हम इसके अंदर इलेक्ट्रॉन बीम यूज करते हैं और ये एज कम्पेयर टू लाइट माइक्रोस्कोप। दूसरी चीज हम लोगों ने ये पढ़ी कि हमारे पास दो टाइप्स होती हैं, एक हमारे पास स्टेम और एक हमारे पास होता है स्कैनिंग इलेक्ट्रॉन माइक्रोस्कोप। अब ट्रांसमिशन इलेक्ट्रॉन माइक्रोस्कोप करते हैं। इलेक्ट्रॉन माइक्रोस्कोप में इलेक्ट्रॉन्स जो होते हैं एक डेंस इलेक्ट्रॉन रीजन से गुजरते हैं, बीम पास होती है और वो स्पेसिफिक जाके हिट करती है और वहां से हमें उसका डिटेल स्ट्रक्चर के बारे में इंफॉर्मेशन पता चलती है। जबकि जो स्कैनिंग इलेक्ट्रॉन माइक्रोस्कोप है उसके अंदर आप अपने सैंपल को गोल्ड या प्लेटियम से जिससे भी हम कोट करते हैं और उसके बाद उसके ऊपर जब इलेक्ट्रॉन बीम यूज होती है तो उसके अंदर जो हमारा स्पेसिमेन है उसके अंदर से सर्टेन इलेक्ट्रॉन्स यानी सेकेंडरी इलेक्ट्रॉन्स एमिट होते हैं और हम उन इलेक्ट्रॉन्स को मेजर करते हैं। तो वो हमें जो स्ट्रक्चर होता है स्पेसिमेन का उसके बारे में इंफॉर्मेशन देते हैं। जो ट्रांसमिशन इलेक्ट्रॉन माइक्रोस्कोप है वो टोटल हमें इंटरनल स्ट्रक्चर्स के बारे में इंफॉर्मेशन देगी, जबकि जो स्कैनिंग इलेक्ट्रॉन माइक्रोस्कोप है फेसेस के बारे में इंफॉर्मेशन देगी। स्कैनिंग इलेक्ट्रॉन माइक्रोस्कोप के अंदर हमारे पास 3D इमेजेस प्रोड्यूस होती हैं, जबकि जो हमारे पास ट्रांसमिशन है उसके अंदर हमारे पास सिंपल जो 2D इमेज होती है वो प्रोड्यूस होती है। दोनों एक्सपेंसिव हैं और दोनों के अंदर ये ड्रॉबैक है कि को डील नहीं कर सकते बिकॉज हमें स्पेसिमेन को कट करके उसके अंदर उसको पूरा हाइड्रेट करके डिहाइड्रेट करके तो फिर यूज करना पड़ता है।
Great, great, awesome. Anaya? Anaya Manzoor.
यस सर।
Fareen, जी
मैंने अभी
जी.
लेट ज्वाइन किया था, मेरा इंटरनेट नहीं चल रहा था।
Okay, you joined late. Fareen, you there?
यस सर।
हाँ, आप एक्सप्लेन कर दें जो फर्स्ट कोई बात है कि यू लाइक यू वांट टू ऐड।
सर सबसे पहले तो हमने देखा था TEM और SEM में कि जिस स्पेसिमेन में से जो इलेक्ट्रॉन बीम जब ट्रांसफर हो जाएंगी सर तो वो होता है TEM और SEM में ये होता है कि जो रिफ्लेक्ट बैक करती है, और SEM जो है वो सिर्फ टोपोग्राफी देता है और जो TEM है वो हमें डिटेल्ड अंदर से सारा कुछ दिखा देता है सर। और सर ये बताया कि दोनों ही एक्सपेंसिव होते हैं और दोनों ही कॉम्प्लिकेटेड होते हैं और दोनों में ही हम लाइव स्पेसिमेन जो है वो यूज नहीं कर सकते।
ठीक हो गया। एंड शफीक इज देयर। शफीक
यस सर।
जी, थोड़ा सा एक्सप्लेन कर दें।
सर इसमें हमने इस लेक्चर में हमने ये देखा है कि लाइट माइक्रोस्कोप इलेक्ट्रॉन माइक्रोस्कोप को इलेक्ट्रॉन लाइट माइक्रोस्कोप की बजाय क्यों यूज करते हैं। इस वजह से कि लाइट जो जो हमें वो मैग्निफिकेशन या रेजोल्यूशन देती है स्पेसिमेन की, उससे ऊपर की रेजोल्यूशन के लिए या इमेजेस के लिए लाइक सेल की इनर डेप्थ डिटेल्स के लिए हम इलेक्ट्रॉन माइक्रोस्कोप यूज करते हैं। तो इलेक्ट्रॉन माइक्रोस्कोप की पेनिट्रेशन ज्यादा होती है एज कम्पेयर टू लाइट पार्टिकल, तो इस वजह से वो हमें हाई रेजोल्यूशन इमेज देती है। तो इसके अंदर भी दो टाइप्स थी, ट्रांसमिशन एंड स्कैनिंग। सो जो ट्रांसमिशन के अंदर हम अपने स्पेसिमेन और पूरा जो मटेरियल होता है उसको इस तरह प्लेस किया जाता है कि जो इलेक्ट्रॉन होते हैं वो स्पेसिमेन के अंदर से पास थ्रू कर जाते हैं। जब वो पास थ्रू करके आगे इमेज क्रिएट करते हैं तो वो हाई डिफाइंड रेजोल्यूशन के साथ एक इमेज दे देते हैं इंटरनल मटेरियल की। बट इन द केस ऑफ स्कैनिंग इलेक्ट्रॉन माइक्रोस्कोप जो इलेक्ट्रॉन बीम होती है उसको हम इस तरह बम्बार्ड करते हैं जो सरफेस के ऊपर जो स्पेसिमेन के सरफेस के ऊपर जाके जब वो हिट करती है तो जहाँ पे वो जो सरफेस के ऊपर जो इलेक्ट्रॉन प्रेजेंट होते हैं वो उसको इमिट करती है और जो उससे रिफ्लेक्शन आती है हम उसको डिकोड करके जो टोपोलॉजिकल जितने भी सरफेस के जितने भी इमेजेस होते हैं वो उसको क्रिएट कर सकते हैं। बट इन द केस ऑफ स्कैनिंग जो रेजोल्यूशन होती है वो ज्यादा अच्छी नहीं होती, लेकिन जो ट्रांसमिशन के केस में रेजोल्यूशन बहुत अच्छी होती है।
Great, great. And who will be the next to explain? Because some
May I, sir?
people have G Azim, please.
सर इसमें इलेक्ट्रॉन माइक्रोस्कोप से सर दो ड्रॉबैक्स हैं. इट हैज टू ड्रॉबैक्स लाइक इट जस्ट प्रोड्यूसेस ब्लैक एंड व्हाइट इमेजेस, वी कैन नॉट आइडेंटिफाई द कलर, बट इन लाइट माइक्रोस्कोप वी कैन डू इट. On second hand, we must need to be a stable thing, and we cannot do this. We cannot use live specimen for identification. So on the other hand, vacuum must be considered. Without vacuum, we cannot use the beam of electron to imagine or to understand the imagery of through the electron microscope. On the other hand, it is the smallest level in we can achieve through the yet possible आने वाले वक्तों में शायद पॉसिबल कि शायद मजीद हम इससे भी छोटी चीज को डिस्कवर कर सकें. I think that's it, sir.
ठीक हो गया. Good.
He knows a lot.
Okay, so first thing first, so scanning electron microscopy, it provides a valuable combination of high resolution imaging, elemental analysis, and crystallographic analysis. So crystallographic analysis, we prefer to use scanning electron microscopy. So and elemental analysis and for high resolution imaging. So the this one is specific for SAM actually. Um, and magnification up to 300,000 x is can be achieved, and identifying crystalline compounds and determining crystallographic orientation of microstructure features as small as 1 micrometer. So this is important for all those who are working with the X-ray crystallography for in that we use in X-ray crystallography we actually uh, make crystalline structure of proteins to visualize the structure of proteins. So to visualize those crystalline structures or crystalline molecules or compounds, we do need scanning electron microscopy. So this is what we do use scanning electron microscopy in day-to-day research. And here, here, this is actually the scanning electron microscopy, how it looks like. Actually, we already covered these things in the videos as well, like the filament and condenser lens, and this has been shown in here, like and condenser aperture, and this the stage, and actually this is for TEM, and this is actually for SEM. So here you can see, and this sample is here, so you already know how to differentiate between the two. So this is not relevant to the scanning actually. This is for TEM, and so components. And there is the scanning electron microscopy. So first of the component is electron column, and the electron column is where the electron beam is generated under the vacuum, and this is the same as vacuum column, and the few focused to small diameter and scanned across the surface of the specimen by electromagnetic deflection coil. The lower portion of the column is called specimen chamber, and the secondary electron detector is located above the sample stage inside the specimen chamber, and the specimen are mounted and secured on the stage. So and the manual state controls are found on the specimen chamber and allow for XYZ movement, so the sample can be monitored. And there is a limit to unaided human eye, so the human eye cannot detect before, and that's the limit for the human eye. And to see microorganism much smaller than zero point 0.1 millimeter, a system required convex lens is needed. And when parallel rays pass through convex lens converged at a point called as the focal length of the lens. So that's important, and hence a lens with shorter focal length has higher magnification power, and the vice versa. And that means that the lens that will have a longer focal length will have lower magnification. Magnification, that means simply means to enlarge an object, and in compound microscope it will be 10x. That 10x and f is equal to the focal length will be equal to 16 millimeter. If that is 40x, so f is equal to 4, and if x 100, so f will be 1.8. So the lower the focal length, the higher the magnification, and so on. So the image produced by the objective lens falls on the eyepiece lens serve as an object. So so that's the image that produced by the objective lens, that will be an object. So and the overall magnification is given as the product of the lenses and the distance over which the image is produced, and that is d into m1 into m2, and that will be divided by 250 millimeter, where d is the projection tube length, and the d will be the tube length, and usually at 250 millimeter, and m1 and m2 are magnification of objective and ocular. And this 250 millimeter, that is the minimum distance of distant vision for 20 by 20 eye. So 20 by 20 is normal eyesight actually. And the what is resolution, and the ability to distinguish between two close objects is called resolution. So if we are able to distinguish between two very similar objects, that means the resolution of our eyes are more. So the magnification should be coupled with good resolution to visualize microorganism, else magnification alone will produce an inconclusive or blurred image. So if magnification is less, or it is not coupled with the resolution, so resolution and magnification, they should be coupled, and if they are not like coherent and to visualize microorganism, so the magnification alone, if we magnify things, if we increase the size of things to be more visible, so the resolution will reduce, and that will produce inconclusive or blurred images. So this is the same, if we drag a picture, it will become blur. So to increase the size of that will become blurred. So magnification is important, but along with that, resolution is also important. Means the resolution is the same like pixels. So pixels is also important. And light microscopy has limits to both its resolution and its magnification. So, in the light microscopy, the optical microscopy, there are limits on both the resolution and the magnification. To overcome this, the electron microscopy was come forward, and the electron microscopy has both high resolution as well as the magnification. So who will explain up till now? Dua is there, Dua?
Yes, sir.
जी अभी तक जो हुआ है ये थोड़ा सा एक्सप्लेन कर दें इस स्लाइड पे.
सर ये सिर्फ रेजोल्यूशन वाली की मैग्निफिकेशन वाली?
दोनों.
सर ठीक है.
Thank you.
वाली स्लाइड जरा कीजिए. सर बेसिकली हमने इसमें मैग्निफिकेशन सबसे पहले होती क्या है. मैग्निफिकेशन किसी भी ऑब्जेक्ट का साइज इंक्रीज करना या किसी भी ऑब्जेक्ट को विजुअलाइज करने के लिए उसको अ काइंड ऑफ जूम होती है हमारे पास मैग्निफिकेशन. जो हमारे पास माइक्रो ऑर्गेनिजम्स होते हैं वो स्मॉलर देन 0.1 मिलीमीटर होते हैं. इनके लिए हमें हाई मैग्निफिकेशन पावर चाहिए होती है इन ऑर्डर टू विजुअलाइज इट. और उसके अलावा यहाँ पे फोकल पॉइंट्स का बताया, फोकल लेंथ का. जब पैरेलल रेज कन्वेक्स लेंस से गुजरती हैं और वो एक पॉइंट पे आके मिलती हैं या कन्वर्ज करती हैं, फोकल पॉइंट बोलते हैं. जितना फोकल लेंथ हमारे पास ज्यादा होगी, मैग्निफिकेशन की जो पावर है वो कम होगी. यानी जितनी फोकल लेंथ हमारे पास कम होगी उतनी हायर मैग्निफिकेशन होगी. जिस तरह नीचे 10x में हमारे पास फोकल लेंथ हमारे पास 16 है तो उसकी 10 टाइम्स है. और अगर हमारे पास फोकल लेंथ 4 है तो उसकी 40 टाइम्स होगी. इसी तरह ही अगर 1.8 पे चली जाएगी हमारे फोकल लेंथ तो हमारी मैग्निफिकेशन 100x तक इंक्रीज हो जाएगी. यानी फोकल लेंथ को डिक्रीज करने से मैग्निफिकेशन इंक्रीज हो रही है. सो नेक्स्ट. सर इसके अंदर बेसिकली दो लेंस होते हैं जो हम यूज करते हैं. एक हमारे पास ऑब्जेक्टिव लेंस होता है, एक हमारे पास ऑक्यूलर लेंस होता है. दोनों लेंस का प्रोडक्ट प्लस जो हमारे पास डी यानी प्रोजेक्शन ट्यूब लेंथ होती है जो कि यूजुअली 250 मिलीमीटर होती है, उसको हम लेकर उसको डिवाइड करते हैं 250 mm से, और फिर हमारे पास एग्जैक्ट मैग्निफिकेशन आता है. 250 जो हम डिस्टेंस लेते हैं वो आइडियल, वो मिनिमम डिस्टेंस होता है जो आपके पास यानी 20x20 आइज के लिए होता है मिनिमम डिस्टेंस, जिसको वो डिस्टिंग्विश कर सकती है. सो नेक्स्ट, रेजोल्यूशन बेसिकली क्या है? मैग्निफिकेशन होता है किसी चीज को जूम करना या किसी चीज का साइज एनहांस करना. लेकिन रेजोल्यूशन क्या है कि अगर दो ऑब्जेक्ट वेरी सिमिलर हैं तो उनको अलग-अलग, उनको एज अ सेपरेट ऑब्जेक्ट देखना, दैट इज रेजोल्यूशन. मैग्निफिकेशन के साथ रेजोल्यूशन का कपल्ड होना बहुत जरूरी है, क्योंकि जब कोई भी चीज मैग्नीफाई होती है तो वो ब्लर हो जाती है, इसलिए उसकी रेजोल्यूशन हाई होना बहुत जरूरी है इन ऑर्डर टू विजुअलाइज इट क्लियरली. सो माइक्रो ऑर्गेनिजम्स को भी विजुअलाइज करने के लिए यही फिनोमेना है. और मैग्निफिकेशन और जो लाइट माइक्रोस्कोप है उसकी रेजोल्यूशन और मैग्निफिकेशन दोनों के अंदर इश्यूज थे, तभी उसको रिप्लेस कर दिया गया इलेक्ट्रॉन माइक्रोस्कोप से, जिसकी मैग्निफिकेशन पावर भी ज्यादा है और जिसकी रेजोल्यूशन, जिसकी वजह से
Exactly.
ऑर्गेनिजम्स इजीली विजुअलाइज हो सकते हैं.
Exactly. Thank you. And next to it is to compare the light microscope with the electron microscope. So first of the things is illuminating source. Light microscope में क्या है? The light is the illuminating source, but in electron microscope, illuminating source is the beam of electrons. So here light is used, here electrons are used. So यहाँ पे in the next point, like what is the second difference? The specimen preparation takes usually minutes to hours, but here the specimen preparation usually takes days. So that's the second difference, but this is a shortcoming in electron microscope as well, because it will take more time if we compare it with the light microscope, the preparation of specimen. And the third thing is we can use live or dead specimen. So that's a positive thing, like we can use a live specimen as well. But here in electron microscope, we only can use dead and dried specimen. We cannot use live specimen. And here the condenser, objective, and eyepiece lenses are made of glasses. Here they are electromagnetic. They are made up of electromagnetic things, and it has low resolving power, but here it has high resolving power, and it is 250 times higher than the light microscope here. It has magnification of 500x and 1 to 1500x, but here in electron microscope the magnification is 100,000 x or 3 to 300,000 x. So here it's a huge difference between light and electron microscope in the magnification as well as in the resolving power. And the objective is 5 micrometer thicker, but here the object is 0.1 micrometer or thinner. Image is colored, so here the image is colored. Here the image will not be colored, that will be black and white, because that we are dealing with electrons only. Vacuum is not required here in this light microscope case, but we have studied that vacuum will be essential in its operation of electron microscope. Here there is no need of high voltage electricity, and here high voltage electric current will be required, and approximately 50,000 volts are above. There is no cooling system required for the light microscope, but here due to high energy consumption and due to the heat production, cooling system will be needed to take out the heat generated by high electric current. So here there will be no cooling system required, and here there will be cooling system required in electron microscopy. And filament is not used here. Tungsten filament will be used to produce electrons. So to have the beam of electrons, we will be having tungsten filament. And here radiation risk is absent, because no radiations are used in a light microscope. and here there is a risk of radiation leakage. Specimen is stained by colored dyes, and that's why we get colored images. Here the specimen is coated with the heavy metals and in order to reflect electrons, and image is seen by eyes through ocular lens, because we will be observing the specimen through the eyes in the lenses. But here the image is received in zinc sulfate fluorescence screen or photographic plate, and it is used to study the across internal structures, but here it is used to in the study of external structure. So internal structures can be studied here, and external structure, ultra structure of cell, and very small organisms can be studied in electron microscopy. And that's it. Who will explain?
May I, sir?
G, please, Azim.
Okay, here we are actually differentiating the light microscope with the electron microscope. In light microscope, the source which we use to illuminate the object is actually light, but in electron microscope, we use electron to illuminate our. And the second thing, then the second difference is specimen preparation takes usually minutes, or we just makes the slide and put it under the light microscope. But in electron microscope, we have to actually cover it with some chemicals, materials, and it takes few days. And third difference is live or dead specimen may be seen like we can use both specimens, live or dead, on the slides, and we can see under the light microscope. But in the electron microscope, we have to use only dead, because because it has to be stable to be focused on the electrons, because we need actually the reflection and deflection of the electrons, so it must be stable. That's why I only use dead organisms to be seen under the electron microscope. Fourth is the condenser objective eyepieces lens are made of glasses. We are actually using the convex lenses. So we are actually using focusing the light onto the focal point. That's why the glasses are used. But in electron microscope, we used electromagnetics to control the beam to give them direction, because electrons can be given direction through the electromagnetism. Okay, in light microscope, it has lower resolving power, uh, uh, as they are using glasses, as we are using glasses in light microscope for magnifying. That's why its resolving power is 0.25 micrometer to 0.3. But in electron microscope, it has high resolving power because we are using the electron beam. That's why it's 250 times higher than the light microscope. In light microscope, our magnification power is 500x to 1500x, but in electron microscope, it's increases to the 100,000 to the 300,000, and it's too much higher than the light microscope. Okay, in light microscope, our organism must be is actually 5 micrometer or thicker, but in electron microscope, the object can be used to be seen is about to be 0.1 micrometer to the, or maybe thinner. Okay, in light microscope, we are actually contacting the direct images through lenses to the object. That's why we can see the exact color of our object. But in the electron microscope, we are actually using the deflection of electrons. That's why we cannot see the exact color of the object. That's why we see the just X-rays of the reflection. That's why it could be in black and white. Okay, but in light microscope, vacuum is not required.
We are not using actually colored dyes here in the electron microscopy.
Yes,
so here, okay.
That's maybe the reason. But in light microscope, vacuum is not required because we are actually using the light. So the light can travel in both vacuum and in non-vacuum spaces. That's why there is no need to be create a vacuum. But for the electrons, in electron microscope, for the electron, we have to generate vacuum, not to disturb the path of the electrons. That's why we need vacuum to let the electrons travel in a straight line. Okay. In the light microscope, we do not need any specific high voltage power. That's why a short 220 voltage would be efficient. But in the electron microscope, it requires a high voltage because it's not a small one. It fits in the large rooms. That's why it requires a lot of electricity to run the whole system of the electron microscope. That's why it requires about 50,000 volts and above. In the light microscope, there is no cooling system. As I have told you, it's just the passing of the light through the object to the eyes. That's why we do not require any cooling system, because our light microscope do not heat up by such amount of light. But in the electron microscope, cooling system is required because a whole generator is being run through the voltage, and electricity is, as we said, that it's about 50,000 volts. So it's generate a large amount of heat. That's why a cooling system is required. In a light microscope, filament is not used because but in the electron microscope, tungsten filament is used to produce electron. In light microscope, we are just using the transfer electron. We can use natural light, and the light generated through the bulb can be used. So that's why no filament could be used. But in light microscope, radiation risk is absent. Radiation is produced by the release of electron or the emission of jumping of electrons from the orbit of the atom. So in light, actually the radiation, there is no risk of radiation, but in electron, an electron could be jumped through the from the atom. That's why there could be a radiation leakage. In light microscope, specimen is stained by the color dyes. Yes, we can use color dyes to see through, because we can see the exact images. That's why there is an experiment we use to detect the sperm in forensics through color dyes. In electron microscope, specimen is coated with heavy metals in order to reflect electrons, because we want to see the trust trough of the images. That's why we have to cover it with such metals which reflect the electrons precisely. That's why we have to cover it with heavy metals. Okay, in a light microscope, image is seen through the ocular lenses. There are two lenses above the microscope which help us to focus on the one point. And the electron microscope, we have our image is seen on the zinc sulfate fluorescent screen or the photographic plate. Okay, in a light microscope, it is used for the study of detailed across internal structure. But our organism, we can observe it about 5 micrometer organisms. But image is received, but in electron microscope, it is used to study the internal organs. As we have to, in the scanning electron microscope, we can observe the external structure of the organisms, and through the transmission electron microscope, we can observe the internal structures of the organisms as well. Thank you.
Thank you. It's well done. And next to it is these are the visuals of the two microscope, the light microscope. The light emit here, and we can and there is a sample here, and we can observe from the lenses. And here is a different system, like the can be visualized on the system here. And so we have a video to compare the electron and light microscope.
Compare light microscopes and electron microscopes. Light microscopes use a light source, so there's going to be a light source on the light microscope. You then place a live specimen onto the stage, so you can use a live specimen. And the light microscope uses lenses. So it uses lenses, the objective lenses and the eyepiece lens. So remember, a light microscope uses a light source. And on a light microscope, there are two lenses, the objective lens and the eyepiece lens. The light on a light microscope travels through the specimen that is on the stage. That is then magnified as it goes through the objective lens, and then it is magnified once more as it goes through the eyepiece lens. Light microscopes can be used to visualize cells and identify the larger structures within the cells. These include the nucleus and chloroplast, for example. You can't see ribosomes using a light microscope. So light microscopes have a lower magnification than electron microscopes, whereby if an eyepiece lens has a magnification of times 10 and the objective lens has a magnification of times 40, you're going to have an overall magnification of times 400. This is lower than the magnification on an electron microscope. Light microscopes have a lower resolution than electron microscopes, whereby you're only able to identify the larger subcellular structures. They are also much cheaper than electron microscopes. It's question time. Attempt these questions to check your understanding. This here is an electron microscope. It requires computers and a lot of space. So this is an electron microscope. An electron microscope can only use dead samples. You cannot use living samples on an electron microscope. Whereas on a light microscope, you can use living samples. So you'd place the sample under the electron microscope. And then what will happen is that the microscope will fire electrons at the sample. So as you can see here, electrons are fired directly onto the sample. So the electron microscope image is going to have a much higher resolution, and a much higher magnification. The higher resolution and higher magnification means that you're able to see smaller structures. So you can actually see inside the nucleus of a cell. You can see the structures of a ribosome, see the structures of a mitochondria too. So remember, electron microscopes have both a higher resolution and a higher magnification, whereby the highest magnification of an electron microscope can be times 1,000,000, and they are obviously going to be much more expensive than light microscopes. So you can use electron microscopes to see the structures of ribosomes and mitochondria. You cannot do that using the light microscopes you have in school. It's question time. Attempt these questions to check your understanding. Electron and light microscope. Press pause to practice using those key words. The answers will follow. Press pause to go through your answers and make any corrections to your mistakes. Press pause to answer the questions. The answers will follow. And if you're stuck, just re-watch the video. Press pause to go through your answers and make any corrections to your mistakes. Press pause to answer this extended response question. The answers will follow. Press pause to go through your answers and make any corrections to your mistakes. Visit kscience.com for more free videos, worksheets and quizzes at kscience.com. And don't forget to like and subscribe. Electron and
Who will explain? जी
क्या मैं
किंजा
कर दूं?
जी फातिमा जी फातिमा बताएं
सर इसमें ये बताया हुआ है कि जो लाइट माइक्रोस्कोप है वो डेड सैंपल्स यूज करता है और उसके दो लेंसेज होते हैं और डायरेक्टली लाइट जो है नेचुरल लाइट को यूज करके वो ऑब्जेक्ट्स को मैग्नीफाई करता है और इसकी मैग्निफिकेशन और रेजोल्यूशन दोनों चीजें लो होती हैं और इसमें हम सिर्फ सेल की आउटर आउटर सेल सरफेस, आउटर सेल देख सकते हैं, इनर भी देख सकते हैं, लेकिन हम उसके न्यूक्लियस वगैरह देख सकते हैं, बहुत स्मॉल ऑर्गेनाइज नहीं देख सकते। और इलेक्ट्रॉन माइक्रोस्कोप में उन्होंने ये बताया कि ये एक बड़ा इक्विपमेंट है, इसके अंदर का हम कंप्यूटर के ऊपर विजुअलाइजेशन करते हैं और इसमें हाई वोल्टेज यूज होता है और इसमें हम इसकी जो रेजोल्यूशन और मैग्निफिकेशन है वो दोनों हाई होती हैं और इसमें हम सिर्फ डेड सैंपल्स यूज कर सकते हैं। और ये बताया हुआ है कि इसमें जो हम इनर जो है सेल की इनर ऑर्गेनाइज है उनको बहुत अच्छी तरह विजुअलाइज कर सकते हैं और इसमें हम ना रेजोल्यूशन भी कर सकते हैं दो डिस्टिंक्ट चीजों में।
ठीक हो गया। सो मूविंग फर्दर,
सर यहाँ पे उन्होंने कहा है कि लाइट माइक्रोस्कोप लाइव यूज करती है और लाइव नहीं, इलेक्ट्रॉन माइक्रोस्कोप लाइव। सर लाइट के लिए तो लाइव और डेड दोनों पॉसिबल है ना सर?
जी जी, लाइट के लिए दोनों पॉसिबल हैं। इलेक्ट्रॉन्स के लिए क्योंकि हमने इलेक्ट्रॉन्स को उसके सरफेस पे डिपॉजिट कराना होता है ना, तो दैट मींस वी हैव टू यूज डेड स्पेसिमेन। सो इलेक्ट्रॉन माइक्रोस्कोपी में डेड स्पेसिमेन यूज होगा एंड लाइट में डेड या लाइव दोनों यूज हो सकते हैं। ठीक है। So if we need to compare SEM and TEM now, that was before this, that was to compare the light and the electron microscopy. Now to compare SEM and TEM. So electron stream is used in both cases. So what difference is there in the electron stream in case of SEM and in case of TEM? So here fine and focused beam is used in electron microscopy, but broad beam will be used in TEM. In transmission, there will be broad beam that is being used, but in SEM that will be fine and fused beam will be used. Image taken is like in SEM, the topographical or surface image will be taken, but in TEM, internal structure will be taken. The image will be of the internal structure. The resolution here, the resolution, if we compare both, the resolution is lower. But in fact it is higher than the light one, but it's lower if we compare with the TEM. Here the resolution is more because we have to go into the detail into the internal structure, so higher resolution will be required. Here the magnification is 2 million times, up to 2 million times, and here the resolution will be up to 50 million times. So we need to know that the resolution as well as the magnification is less if we compare the SEM with TEM. So TEM has higher resolution as well as higher magnification. And image dimension here, that will be 3D. An image dimension that is taken with TEM is 2D. And sample thickness, that thin and thick sample are okay. So if we take thin samples or thick samples, so that's okay for SEM. But for TEM, for transmission, ultra thin samples are used. And penetration of samples occur or not. So here no penetration of the beam into the sample, but here the penetration will occur. Sample restriction, that is less restrictive. Here that is more restrictive. And sample preparation is less preparation required. Here more preparation will be required, more time will be required. And for the cost, the SEM is less expensive because it's less energy in context with resolution, in context with magnification, and here the sample preparation, and so this is less expensive. But this TEM is more expensive. Speed, for the speed, this is faster. The results will become like that's come sooner than the TEM, and this will be slower because it has to go into more details with high resolution and with higher magnification. So that will be slower. operation, how it will be done, like easy to use, is easy to use, and here it's more complicated and requires training. Who will explain?
सर मैं कर दूं?
G, please.
सर बेसिक इसमें हमने कंपैरिजन किया स्कैनिंग इलेक्ट्रॉन माइक्रोस्कोप का और ट्रांसमिशन इलेक्ट्रॉन माइक्रोस्कोप का. सबसे पहले इफ वी टॉक अबाउट इलेक्ट्रॉन बीम, तो सेम के अंदर फाइन फोकस्ड बीम होती है. वो इसलिए क्योंकि हमने सिर्फ उसके सरफेस पे उसे बीम को फोकस कराना होता है. वहां से जो इंटरनल इलेक्ट्रॉन्स होंगे हमारे स्पेसिमेन के अंदर वो बाहर आ जाएंगे और उसको हम यूटिलाइज कर लेंगे. लेकिन यहाँ पे ट्रांसमिशन इलेक्ट्रॉन माइक्रोस्कोप के अंदर हमारे पास ब्रॉड बीम यूज होती है. जो इमेज होती है हमारे पास सेम के अंदर वो टोपोग्राफी इमेज होती है, यानी वो सिर्फ आउटर सरफेस एरिया की मैग्निफिकेशन होती है. लेकिन जो टेम होती है वो हमें इंटरनल स्ट्रक्चर के बारे में इंफॉर्मेशन देती है. इफ यू टॉक अबाउट रेजोल्यूशन एंड मैग्निफिकेशन, जो हमारे पास टेम होती है उसकी रेजोल्यूशन और मैग्निफिकेशन दोनों लो होती है एज कम्पेयर टू ट्रांसमिशन इलेक्ट्रॉन माइक्रोस्कोप, बिकॉज उसकी रेजोल्यूशन और उसकी मैग्निफिकेशन ज्यादा होती है. जो हमारे पास इमेज डायमेंशन है सेम के अंदर डी इमेज प्रोड्यूस होती है और ट्रांसमिशन के अंदर हमारे पास 2D इमेज प्रोड्यूस होती है. सैंपल थिकनेस के अंदर सेम के अंदर क्योंकि हमने सिर्फ सरफेस एरिया को चेक करना होता है, सैंपल्स बोथ कैन बी यूज्ड. जबकि ट्रांसमिशन इलेक्ट्रॉन माइक्रोस्कोप के अंदर हमें अल्ट्रा थिन सैंपल चाहिए होते हैं बिकॉज इलेक्ट्रॉन उसके पेनिट्रेट करना होता है. और पेनिट्रेशन जो होती ट्रांसमिशन इलेक्ट्रॉन माइक्रोस्कोप के अंदर होती है, स्कैनिंग इलेक्ट्रॉन माइक्रोस्कोप के अंदर नहीं होती. सैंपल रिस्ट्रिक्शन जो हमारे पास सेम का सैंपल लेस रिस्ट्रिक्टिव होता है एज कम्पेयर टू ट्रांसमिशन इलेक्ट्रॉन माइक्रोस्कोप. सैंपल प्रेपरेशन की अगर बात करें तो अगर हम स्कैनिंग इलेक्ट्रॉन माइक्रोस्कोप को देखें तो इसका सैंपल को प्रिपेयर करने में यानी लेस टाइम लगता है एज कम्पेयर टू ट्रांसमिशन इलेक्ट्रॉन माइक्रोस्कोप. कॉस्ट सेम की कम है एज कम्पेयर टू ट्रांसमिशन इलेक्ट्रॉन माइक्रोस्कोप. इजी टू ऑपरेट है हमारे पास स्कैनिंग इलेक्ट्रॉन माइक्रोस्कोप टू यूज है. लेकिन अगर हम ट्रांसमिशन इलेक्ट्रॉन माइक्रोस्कोप को देखें तो वो उसकी स्पीड स्लो है बिकॉज उसने कॉम्प्लिकेटेड स्ट्रक्चर्स को विजुलाइज करना होता है, तो उसका टाइम ज्यादा लगता है विजुलाइजेशन में. जबकि सेम के अंदर सिर्फ आउटर स्ट्रक्चर को विजुलाइज करना होता है या उसकी सरफेस एरिया को विजुलाइज करना होता है, तो उसके लिए कम लगता है.
Gee, that's good. One more thing, that's they are less restrictive and they are more restrictive. In this case, they are the electron microscope. They are more sensitive to vibration and electromagnetic fields, and therefore they are housed. They are kept in isolated rooms. The electron microscopes. But for in the case of the STEM, it requires higher resolution and it requires higher magnification, so they are more energy intensive, and so they need to maintain high voltage. So the voltage should be kept maintained at a constant at a constant range, so it is more restrictive, and this is less restrictive, so it's more this is more sensitive actually, and the rest is okay. That's good. And okay, if we can see the diagrams of SEM and TEM. So this is SEM and this is TEM. So if we compare both, the electron source is common for both. The anode, so this magnifies the beam. Condensers are there, and after condenser there is the specimen kept in SEM, in TEM, and here the other condenser lens is used. So here the objective lens, because there is the object, so this will be the objective lens. This is the condenser lens, this will be the objective lens. But here in case of SEM, both will be the condenser lens. After that, this is the intermediate lens, and here the objective lens, because the object is now here, the specimen is here, and now after that back scattered electron detector, and after that they are detected the X-rays and secondary, and the electron detectors, are X-ray detectors are used. And here the fluorescent screen here directly that will detect the specimen. So That's what is the difference between the two. So in one case, the light will, the electrons will, the source will go through the specimen, but here it will reflect from the specimen, and it will, the image will be taken. And that's it.